This is the second lecture of the three-lecture series regarding DNA metabolism. We will cover DNA repair in this lecture. DNA repair and mutations. Chemical reactions and some physical processes, for example UV or gamma rays, constantly damage genomic DNA. The majority are corrected using undamaged strand as a template. Some base changes escape repair, and an incorrect base serves as a template in replication. The daughter DNA carries a changed sequence in both strands. And accumulation of mutations in eukaryotic cells is strongly correlated with cancer. Most carcinogens are also mutagens. There are thousands of lesions per day. Lesions are unrepaired DNA damages, but only one in every 1,000 become a mutation. This is thanks to DNA repair. The human genome contains genes for more than 130 repair proteins. Ames test for carcinogens. The Ames test was developed by Bruce Ames. It indicates the mutagenic potential of a compound. A strain of Salmonella typhimurium having a mutation that inactivates an enzyme of the histidine biosynthetic pathway is plated on a histidine-free medium. Few cells grow in A. The few small colonies of S. typhimurium that do grow on a histidine-free medium carry spontaneous back mutations that permit the histidine biosynthetic pathway to operate. Three identical nutrient plates, B, C, and D, have been inoculated with an equal number of cells. Each plate then receives a disc of filter paper containing progressively lower concentrations of a mutagen. The mutagen greatly increases the rate of back mutation and hence number of colonies. The clear areas around the filter paper indicate where the concentration of mutagen is so high that it is lethal to the cells. As the mutagen diffuses away from the filter paper, it is diluted to sublethal concentrations that promote back mutation. Mutagens can be compared on the basis of their effect on mutation rate. Because many compounds undergo a variety of chemical transformations after entering cells, compounds are sometimes tested for mutagenicity after first incubating them with a liver extract. Some substrates have been found to be mutagenic only after this treatment. Types of DNA damage. Mismatches arise from occasional incorporation of incorrect nucleotides. Abnormal bases arise from spontaneous deamination, chemical alkylation, or exposure to free radicals. Pyrimidine dimers form when DNA exposed to UV light. Backbone lesions occur from exposure to ionizing radiation and free radicals. These are the different DNA repair systems in E. coli. We will discuss some but not all of the repair systems in E. coli. Mismatch repair and methylation. Methylation of DNA strands can serve to distinguish parent, which is the template, strands from newly synthesized strands in E. coli DNA, a function that is critical to mismatch repair. Demethylation occurs at the N6 of adenines in the GATC sequence. Remember, by default, we communicate sequences from 5' to 3'. This sequence is a palindrome. Thus, the opposite strand is also methylated. Following replication, only the parent DNA is methylated. This means that any mismatch error during replication must be present in the unmethylated DNA. The methyl-directed mismatch repair system will cleave the unmethylated strand in the initial part of the repair process. There is a limited amount of time before the unmethylated strand is methylated, and the ability to repair the mismatch is lost. Methylation-directed mismatch repair in E. coli, the early steps. Recognition of the sequence GATC and of the mismatch are specialized functions of the MUT-H and MUT-S proteins, respectively. The MUT-L protein forms a complex with MUT-S at the mismatched DNA. DNA is threaded through this complex such that the complex moves simultaneously in both directions along the DNA until it encounters a MUD-H protein bound at the hemimethylated GATC sequence. This movement makes a DNA loop, and MUD-H cleaves the unmethylated strand on the 5' side of this G sequence. The mismatch could be 1,000 base pairs away from GATC, a complex consisting of DNA helicase II and one of several exonucleases then degrades the unmethylated DNA strand from that point in a 3' to 5' direction towards the mismatch. The removed sequence is replaced using DNA polymerase III and DNA ligase. Methylation-directed mismatch repair in E. coli, the latter steps. The combined action of DNA helicase II, SSB, and one of 4 different exonucleases removes a segment of the new strand between the MUD-H cleavage site and a point just beyond the mismatch. The exonuclease that is used depends on the location of the cleavage site relative to the mismatch, as shown by the alternative pathways here. The resulting gap is filled in, shown by dashed lines, by DNA polymerase III, and the nick is sealed by DNA ligase. Repair by base excision repair pathway. Here we're looking at what happens when cytosine spontaneously deaminates to uracil. The deamination of C to U occurs at a frequency where its correction is necessary to inhibit the accumulation of mutations. Moreover, uracil does not belong in DNA. In step 1, DNA uracil glycosylase recognizes a damaged base and cleaves between the base and deoxyribose in the backbone. In step 2, an AP endonuclease cleaves the phosphodiester backbone near the AP site. In step 3, DNA polymerase I initiates repair synthesis from the 3' hydroxyl at the nick site, removing with its 5' to 3' activity, and replacing a portion of the damaged strand. In step 4, DNA polymerase I dissociates, and the remaining nick is sealed by DNA ligase. Note that the DNA glycosylase initially cleaves the N-glycosyl bond between the sugar and the base. The entire nucleotide and sometimes around the AP sites are ultimately removed. Nucleotide excision repair in bacteria and humans. The general pathway of nucleotide excision repair is similar in all organisms. In step 1, an XC nuclease binds to DNA at a site of a bulky lesion and cleaves the damaged DNA strand on either side of the lesion. XC nucleases are UV-specific excision endonucleases. In step 2, the DNA fragment, a 13-mer in E. coli and a 29-mer in humans, is removed with the aid of a helicase. In step 3, the gap is filled by DNA polymerase, and in step 4, the remaining nick is sealed with DNA ligase. Lesions include pyrimidine dimers, 6,4 photoproducts, both caused by UV light, and benzopyran guanine, which is caused by cigarette smoke. What happens if there's no template to correct for an error? Here are 2 scenarios where this can occur. If the replication fork encounters an unrepaired lesion, as shown on the left, or a strand break, as shown on the right, replication generally halts and the fork may collapse. Alternatively, a lesion is left behind in an unreplicated single-stranded segment of DNA, as shown on the left. A strand break becomes a double-strand break, as shown on the right. In each case, the damage to one strand cannot be repaired by mechanisms described earlier, because the complementary strand required to direct accurate repair is damaged or absent. There are 2 scenarios for repair: recombinational or error-prone repair. Error-prone repair occurs when there are many lesions present. This process uses DNA polymerase V, which can replicate over many lesions. Unfortunately, the process is inaccurate and many mutations result.